Bismillahirrahmanirrahim. So far we have discussed volume 2, volume 3 and volume 4, and among those volume 2 belongs to gram-negative organisms, 3 and 4 belongs to gram-positive organisms, and volume 1 also belongs to gram-negative organisms. But I told you that all the other organisms which couldn't be grouped in volume 1 and 2, and they were separated into another volume, which is the volume 5. So basically volume 5 includes those gram-negative organisms which are not uh, which were not placed in the volume 1 and volume 2. So basically this phylum it contains variety of different phyla, but we'll discuss a few important ones. And these phylums, they include Planktomyces and Chlamydia, Spirochaetes, and Bacteriology, and Fusobacteria. So these 5 will be discussed in this lecture. First, we start from Planktomyces. So the Planktomyces, they are the gram-negative budding bacteria, and they are known to blur the definition of what bacteria are. Means on what grounds we say any cell as a bacterial cell, because we know that bacteria, they are prokaryotes, and prokaryotes, they do not have a complete nucleus, or nuclear membrane is absent, and membrane bound organelles are absent. So basically this group is unique in that sense that in this group there are some members which have some structures that are similar to the eukaryotic species. For example, if we say that as per their 16S DNA analysis, they belongs to bacteria. But if you look at the cell and the internal structure of the cell, so they resemble archaea in the makeup of their cell wall. Means they do not have peptidoglycans in their cell wall, which is one of the major property of a bacterial cell wall. So they do not have bacterial peptidoglycan. So they resemble archaea, and plus they may have some organelles that resemble the eukaryotic nucleus. So you can see that one of the species we will discuss, Gameta globi, Gameta, Gameta obscuri globus. There is a species which is known to have a double internal membrane around its nucleus, which is present in the eukaryotic nucleus. So definitely that's why they are known to blur the definition of what bacteria are. Although their DNA analysis and its resemblance places them into the bacterial branch, but they also have some of the very important properties, and that are the characteristics of other groups like eukaryotes and the archaea. So one of the genus of Planktomycetes is the genus Planktomyces, and they are the aquatic bacteria which produces stalks like Colobacter. So there is another organism which is, I told you just now, that Gameta obscuri globus. It has a double internal membrane around its DNA, which resembles a eukaryotic nucleus. So this organism can be served as a model for the studies regarding the evolution of a nucleus, or the origin of a complete nucleus. in the same way that we discussed about mitochondria, origin of chloroplast, right? So this organism can be used as a model organism because it contains almost a similar eukaryotic nucleus, and it's similar in that sense that it has a double internal membrane around its DNA. This is very unique in this sense. Then we have another phylum which is known as Chlamydia and Chlamydia phylum, Chlamydia. So this phylum is genetically similar with bacteria that again do not contain peptidoglycan in their cell walls, and we have compared Chlamydia in I think volume class 1 of volume 2, alpha Bifidobacteria. Right, in alpha Bifidobacteria there are Rickettsia which are placed in alpha Bifidobacteria, so they were compared with Chlamydia. And I also showed a slide when I was giving a lecture on viruses. What is the difference between viruses, Chlamydia, and the Rickettsia, because all are obligate intracellular parasites. So they grow intracellularly within host cells. And there are two important genus in this phylum, which are Chlamydia and Chlamydophila, and they do not require insects or ticks for transmission because unlike Rickettsia requires, or rickettsial transmission requires insects or any vector including ticks and insects, but its transmission is not through the insects or ticks. They are transmitted by airborne respiratory routes or the interpersonal contacts. And secondly, they can be cultivated artificially in the laboratory animals or in the cell culture, because you cannot cultivate them on the agar base because the same reason that because they are intracellular parasites. So definitely you need to have cell culture, or you need to grow them in the yolk of embryonated chicken eggs. So you can use them both. Chlamydia and Chlamydophila, and they have a unique life cycle patterns we'll discuss. And at first we'll discuss about Chlamydia. So one of the pathogenic strain of Chlamydia is the Chlamydia trachomatis, and this is the best known pathogen of the group, and it is responsible or the main cause of trachoma, and the trachoma is the most common cause of blindness in humans in less developed countries. So Chlamydia trachomatis is one of the major cause of blindness in humans. Then there are two members of Chlamydophila, which are the well-known pathogens, and they include Chlamydophila psittaci, which causes the psittacosis, what is also known as ornithosis. And another member is Chlamydophila pneumonia, which also causes a mild form of pneumonia. So I think if we, I have discussed it before, but recalling it again, that if I give you some names, abbreviated names, then and ask you to de-abbreviate them, then I should, then you should be able to do it. Like for example, if I give you some names like S pneumoniae, M pneumoniae, H pneumoniae, and C pneumoniae, then I expect you that you can differentiate. But what does it mean by C pneumoniae? It means that Chlamydophila pneumoniae. And what does it mean by S pneumoniae? They have unique life cycle pattern because this will be discussed for the very first time, such life cycle pattern. They do not divide by budding, right? They display a unique life cycle which takes about 48 hours to complete. So in the entire life cycle, there are two things, or the life cycle based on two forms, two cellular forms. One is known as the elementary body, and the other one is known as the reticulate body. You can see it here as well. So basically, there are two. This is the cycle of two life forms: the elementary body and the reticulate body. So basically, the infectious form is the elementary body, as you can see that Chlamydia and Chlamydophila both display this type of life cycle. So that elementary body is infectious form and it attaches to any host cell. Then it gets internalized and phagocytized and in the form of a vacuole. And then this vacuole, within this vacuole, in this articulate body, it matures itself and it forms a bigger structure, which is known as reticulate body. And once it is transformed from elementary body to reticulate body into any host cell, it starts replicating itself, dividing successfully into multiple copies. Multiple reticulate bodies are formed, and then there comes a stage where these reticulate bodies, they can start converting themselves into elementary bodies, and again, and this will be released and the host cell dies. Right, and again elementary bodies, they are ready to infect a new cell. So this is the life cycle which is unique and we have not discussed it anywhere else. So you can remember these two groups with this reference of life cycle. So after Chlamydia, we have spirochetes, and you probably have seen spirochetes in your lab session as well. So they have coiled morphology, but they are different from we have discussed spirals. They are different spirals. There's spirals, and they are the tight coils, right? So spirochetes, they have coiled morphology, and they are different from the group Spirulum, right? And their genetic makeup is different. Their morphology is somehow similar, but you can see a visible difference, and it was shown in that slide as well, in which I was discussing the group Spirulum. So it resembles a metal spring, and I told you, they're more tightly coiled, and secondly, they are motile, but their motility is very distinct. That is because of the presence of axial filaments, which are present between the outer membrane and the internal structure of the cells. So the twisting of these filaments inside this space, it is responsible for the movement of these spirochetes. So they have a motility which is by means of not the extra exoflagella, but it is because of the endoflagella, internally present flagella. They are mostly found in the human oral cavity, and secondly, there is or extraordinary location for spirochetes that they are found on the surface of the protozoa, which is found in termites. Cellulose digesting protozoa found in termites, where these spirochetes, they function as substitute for flagella. They serve as flagella for that protozoa. So this is I found something very unique or different, right? That how symbiotically they maintain themselves, serving as a flagella for the protozoa. So one of the example of spirochetes is the genus Streptomyces, and one of the pathogenic species is the Treponema pallidum, which causes syphilis. And then we have within this phylum Spirochaetes, we have some other important genus like other than Treponema, we have Borrelia and we have Leptospira. So Borrelia is responsible for causing relapsing fever and Lyme disease, and these are the diseases which are transmitted by ticks or lice. So and then we have Leptospira. Leptospira basically the genus member of this genus, they are responsible for causing leptospirosis, and it is a disease which is spread to humans and by water contaminated with leptospira. Basically, these leptospira species, they are excreted in urine of animals like dogs, rats, and swine. So domestic dogs and cats, they are routinely immunized against leptospirosis, which is usually common in those countries where the trend of keeping pets is common. So if they are routinely immunized, then this can be easily controlled. And there is another phylum which is known as Bacteriology, and they are the most common inhabitants of your gut, and they are anaerobic bacteria because definitely gut is that environment where only anaerobes or the facultative anaerobes can survive. So there are several genera, but we will discuss Bacteroides and Cytophaga. Bacteroides, as you can see, I told you that they are the human intestinal tract inhabitants, and their number is reaching approximately 1 billion per grams of the species. So alone Bacteroides, it is comprising this much ratio. So this is one of the most abundant organism found in your intestine. So it is also it can also resides in your gingival crevices. So because it provides the anaerobic environment which favors the Bacteroides species. They are non-motile and non-sporing, and once they are spread from the site of injury, from during surgery or from any wound, so they may cause an infection which is known as peritonitis, or also known as also characterized by perforated bowel. So they are frequent cause of peritonitis. Another important genus belonging to Bacteroides phylum is Cytophaga, and they are important in the degradation of cellulose and chitin, and they are mostly found in soil. So, and they display gliding motility, and this is a feature which is remarkably known for the Cytophaga, right? Then we have Fusobacteria, as its name indicate, that the word fuso is representative of a spindle form things. So as you can see that these are the cells which is not showing the blunt ends, rather spindle ends. So this is something unique regarding the morphology of bacterial cell, right? So they are also anaerobes. It's another phylum of anaerobes. They are often pleomorphic, means they can different members can acquire different shapes, but mostly their name suggests that they may be spindle shaped. The ends may be spindle shaped. So they're most found again. They are anaerobes, so they are mostly in the gingival crevices of the gums, and they are responsible for some dental abscesses. So these are the Fusobacteria, which are known for their spindle shape.